We left off looking at this cross-section of early secondary growth, where we still have the epidermis, and we have formed the vascular cambium, and it has just started making secondary tissues. Now we're going to jump into full-on secondary growth. I think this will be about a year in, so we're looking at a slice that will look similar to this diagram here. So this is later in secondary growth. We now have our crushed epidermis up here sloughing off, and we have the formation of our periderm starting out. Most of the periderm is going to be cork cells. There will be many more layers of cork cells produced than there will be layers of phyloderm. So up here you see all cork cells. They're dead. They have no contents. They start getting suberized, so filled with wax. Right below that, these layers of dead empty cells, you see these kind of squished together little densely packed cells. That is going to be our cork cambium, which is making those cork cells. And just below the cork cambium, there might be some phyloderm or phylogen that has started to be produced. Okay, so that is the outer layer of secondary tissues. In here, we now have several layers of our secondary xylem forming and a thicker layer of secondary phloem. So to find the the vascular cambium, you would look in between those. So the xylem in the stains that we'll see in images is usually stained like a pinkish or reddish color due to the secondary walls. If you are doing this at home and you're using toluidine blue, remember that those secondary walls stain like an aqua blue color. Sometimes they look a little gray. So here we have our secondary xylem cells. Those secondary xylem cells also tend to be filled with larger vessel elements, right? That's part of how we can find the xylem, is look for those large empty cells with thick walls. So we have secondary xylem here. Our primary xylem is down here in these little pockets where they used to be formed in these bundles, but now we're just forming continuous rings. So we can follow that secondary xylem, and the newest secondary xylem that's been made is right here. And just above that is this layer of tiny, tiny little bright blue cells. That's our vascular cambium. So our vascular cambium will make xylem, secondary xylem to the inside, and to the outside of the plant it will make secondary phloem. So up here, this is all secondary phloem. You can see these bundles of fibers here. That shows you where the primary phloem was. So right next to those bundles of phloem fibers, you would find primary phloem cells. Not super important that you know that, but if you're wondering, where did that primary phloem go? You can look for those bundles of fibers. It's not present in all plants, but it is present in some plants, like this one we're looking at right here. And then we still have some cortex here. So this cortex will keep getting converted into cork cambium, which will then continuously produce more cork and phyloderm. Okay, so we are early secondary growth here. We formed a few layers of secondary xylem and secondary phloem, but we haven't really hit like multiple years of growth yet. We don't have distinct growth rings. So let's compare these two. I'm just going to move myself out of the way for a second. So we have pith down here at the bottom. We have primary xylem here and primary xylem here. We have secondary xylem starting to form in this one. Notice that none of these cells have secondary walls formed yet. They all have stained the same color. We're staining the primary wall here. There's no real secondary wall visible in that secondary xylem. However, you can see it in the primary xylem. In these, there's this little like peachy color to that secondary wall. In this one, we do see a start to change in the color. So we're starting to form those secondary walls and maturing that secondary xylem. Here we have our vascular cambium, and then above that will be a little bit of secondary phloem here, much more secondary phloem in this one. Our primary phloem is here next to these phloem fibers. Primary phloem in this one is hard to distinguish where it is. It's probably still mostly primary phloem. We have cortex, and in this one we just have all cortex and epidermis. We don't have any cork cambium yet. Here we've started to form our first layers of periderm. So early secondary growth on your left, just starting out, and then maybe starting to be about a year in on the right. So now we can look at this again in long section, looking at these stages as they occur as we're transitioning into older secondary growth. So still, we're not quite at a year here. Now let's jump forward in time and look at a 3-year-old stem. So here we have a cross-section that we've then zoomed in on, and this is, I think, a different plant species than the one we were looking at. They might all be Tilia, but I can't remember. Okay, so in this one, we have our pith here in the center. We have little pockets of primary xylem here, but you can't really see them very well. They would be just kind of dipping into the pith a little bit. And then we have multiple layers of secondary xylem. And you can distinguish these layers as distinct rings, which is cool because that helps us age these plants that go through secondary growth. So why does it form in these distinct rings? We can look over here on the right and see why those rings sort of form. If you look right where the sort of line of the ring is, you start to see all of these densely packed tiny vessel elements. So these densely packed tiny vessel elements form in times where there's very little water, or it's very cold, so there's very little water. So this is the end of the growing season, once you start to go dormant. So then the tree goes dormant. This is an angiosperm, a flowering plant. It's most likely going to lose its leaves. Not necessarily, but most likely going to lose its leaves in the wintertime. And then it'll overwinter, kind of cease growing, trying not to transport too much water because that water can then freeze inside its tissues. Then in the spring, puts out a flush of new leaves, starts pumping water really fast, makes huge cells so that it can do that. So that's what these vessel elements are, this spring wood that forms. So this is sometimes called spring wood or early wood. It's the large vessel elements that form when the plant starts the growing season. Then we progress through the growing season and we make smaller vessel elements because there's less water and we're starting to get maybe some variation in temperature where we're going to be worried about water freezing within our cells. So if we make them smaller, we're less likely to get air pockets, embolisms from that. So we go from large early wood cells to small late wood cells. So this would be like spring into fall or summer into fall. And then at the very end there, that's where we stop growing for the season. So each of those represents a growing season or a year. And then we can count how many of those have happened to see approximately how old this plant is. So that's our secondary xylem. We call that wood. Secondary phloem is out here, and it's very distinct once you get to these older stages of the tree because it has tons of phloem fibers. So all of this pink stuff within the secondary phloem is all phloem fibers. The blue stuff in there, very little of it, but that's the living cells that are going to be actively conducting. So if we take a closer look at that over here on the right, you can see tons of fibers. So this phloem tissue is still pretty tough. You can imagine if you just had a column of tough woody cells surrounded by some like floppy living cells, and then with tough woody cells again on the outside, what's your support for that layer of really tender cells that are transporting highly valuable stuff, right? So that's where these phloem fibers come in, is they support and protect this layer of living cells. This is really between the vascular cambium and the cork cambium. Those are the living cells within a tree. And then the leaves, right, and the growing tips. But most of a tree is dead. You have, mostly in the secondary phloem, you have phloem fibers, and the entire secondary xylem is dead, except right here, and this is going to be on a future slide, so I'll just talk about it later, but you have rays of living cells that travel between these, because you still need to transport things back and forth between the xylem, the phloem, and the rest of the plant. Okay, so here is, much like the root, a look at the sort of stages that you're going through. So again, here we would be in that growing root tip or shoot tip, sorry. So many words. Going through the growing shoot tip, where we still haven't made our primary tissues, we just have our primary meristems. They are then going to make our primary tissues, which we can see forming here into these mature cells. Xylem is always on the inside, phloem's on the outside. Between those, we would have some Vascular Cambium, and then in between them, we would have some intervascular cambium, which is labeled here in C. Pith is on the inside, cortex is on the outside, and then the epidermis is our outermost layer. Here we start to see we're getting a little bit older. We have connected our cambium here. We have a full vascular cambium that's now made some secondary tissues. We still have our cortex and our epidermis because our cork cambium hasn't really fully formed yet. We don't see that until a little bit later in secondary growth where we start to form our periderms. And remember, it's the cortex that is going to form the cork cambium. Okay, so now we get to the rays, which I'm going to talk about this in the next video because we're at 11 minutes.